For any assignment-related queries, call us at +1 678 648 4277. You can mail us at info@nursingassignmenthelp.com or reach us at https://www.nursingassignmenthelp.com. Molecular Biology Assignment Help. Question 1. You are a graduate student in a lab studying the human protein YCG1. From a previous graduate student's work, you know that there are different levels of YCG1 in different tissues, and you are interested in understanding the mechanisms determining the levels of YCG1 in these different tissues. You decide to start by comparing levels of YCG1 mRNA and protein in liver cells and kidney cells. 7a. How would you go about purifying RNA poly transcripts from each cell type, both pre-mRNA and mRNA? And what assay would you use to look at YCG1 mRNA levels? First, you must separate the nuclear pre-mRNA from the cytoplasmin mRNA. Then, to purify pre-mRNA or mRNA, use an oligo-DT column. Oligo-DT will bind to the poly-A tail of the transcript. You could use a northern blot or RT-PCR to assay mRNA levels. 7b. How would you determine levels of YCG1 protein in the different cells? Precipitate the protein from each of the different kinds of cells, run on an SDS-PAGE gel and do a Western blot. If you don't have antibodies to the YCG1 protein, then you can tag it, for example HA, MYC, flag, etc., and use antibodies that recognize the tag. Nursingassignmenthelp.com. Curiously, you find that the levels of YCG1 pre-mRNA and mRNA are the same in the two tissues, but that there is 8 times more YCG1 protein in kidney cells than in liver cells. You also notice that the mRNA from the kidney cells is longer than in the liver cells, but that the pre-mRNA is the same length. 7c. You suspect that the difference in mRNA length is due to alternative splicing. What experiment would you do to test this hypothesis? Make cDNA, isolate the mRNA from the cytoplasm using oligo-DT, add oligo-DT primer, which will bind to poly-A tail, RT, and dNTPs. Hairpin acts as primer for second strand. Get rid of RNA, for example alkali, add DNA poly and dNTPs to get second strand synthesis, and add S1 nuclease to cut hairpin. You can get both length and sequence information from cDNA. You determine that the difference in mRNA length is not due to alternative splicing. Your next hypothesis is that the YCG1 mRNA poly-A tails are different lengths in the two cell types. 7d. How could the difference in poly-A tail length lead to different levels of protein in the two tissues? Nursingassignmenthelp.com. The poly-A tail is important in getting efficient translation of the mRNA. It could be that in liver cells, the YCG1 mRNA poly-A tail is very short, and so translation does not occur very efficiently, and thus there isn't a lot of YCG1 protein. In contrast, the YCG1 poly-A tail in kidney cells could be longer, and thus translation occurs frequently and there is more protein. The mechanism behind the importance of the poly-A tail in translation. The poly-A binding protein coats the poly-A tail, and the poly-A binding protein binds the initiation factor EIF4F. This provides a connection between the poly-A tail of the mRNA and initiation of translation. 7e. You would like to test your hypothesis. Based on your knowledge of the role of the poly-A tail in translation, what protein would you propose to delete to test your hypothesis? You would delete the gene that codes for the poly-A binding protein. In this environment, the YCG1 protein levels in kidney and liver cells should be equal. Question 2. You are studying the regulation of the gene encoding release factor 3, RF3, from a recently isolated single-cell eukaryote. You first assess the abundance of the RNA encoding the RF3 as well as the RF3 protein. You obtain the following results. NursingAssignmentHelp.com 8. At what stage is RF3 synthesis regulated by amino acids? RF3 synthesis is regulated during translation because transcription levels of RF3 mRNA are identical, but Western protein amount differs. To gain insights into the mechanism of control, you want to isolate mutants that alter the control of RF3 expression. You only have the sequence of the first 75 bases of the mRNA available to you, but the first August is present in this region. NursingAssignmentHelp.com. You make 2 fusions to LAX. One precisely places the August of LAX in the same place as the first August of the RF3 gene. The second uses a convenient restriction site located 250 bases into the mRNA, see below, but you cannot tell whether it is in the same reading frame as the first August or not. You test each fusion for beta-galactosidase activity with high and low amounts of amino acids present in the growth media. You obtain the following results. 8b. What can you conclude about the mechanism of control of RF3 expression based on these results? NursingAssignmentHelp.com. One can conclude that the first 200 bases of RF3 are required for regulation of the RF3 mRNA. You use the second construct shown above to screen for mutants that are misregulated. You have some difficulty but eventually identify 3 mutations that alter RF3 expression and call them RRT1, RRT2, and RRT3. They have the following characteristics. RRT1 cis, constitutive RRT2 trans, uninducible RRT3 cis, uninducible. Having identified 2 cis-acting mutants, you decide to sequence the rest of the WT and mutant mRNAs. All instances of either start, August, or in frame stop codons are shown. NursingAssignmentHelp.com. 8c. Using the above information, explain the phenotypes of the RRT1 mutation. Propose a model to explain the RRT3 mutation. RRT1 mutant lacks the ORF of the WT, which is required to negatively regulate the translation. The mutant is constituted because the ribosome does not react to the AA levels. It binds August of LAX regardless, no negative regulation. RRT3 mutant lacks the stop codon in the ORF. The ribosome binds the August of the ORF and starts going all the way through LAX, which is most likely out of frame since no product is produced. Uninducible August of LAX is not recognized because it's inside an ORF. NursingAssignmentHelp.com. To be sure that the phenotypes that you observe are relevant to the normal regulation of RF3 expression, you make the same mutations in the normal RF3 gene, without the LAX fusion, and find the following results. You also sequence the entire wild-type mRNA and find that there is only 1 August in the entire coding region. All AUGs and stop codons are shown below. NursingAssignmentHelp.com. Finally, you also clone the gene that is mutated in RRT2. You find that it encodes a tRNA that is normally expressed at low levels. Interestingly, the anticodon of the tRNA recognizes the UAG stop codon. 8d. Given that reduced amino acid levels result in substantial reductions in the level of translation, and therefore an increase in the amount of available RF3, propose a model to explain the regulation of the expression of the RF3 protein. RF3 is required to stop translation for a stop codon to work. When AA levels are low, RF3 is around to stop translation at UAG, and little RF3 is produced. When AA levels are high, there is little RF3 around and TR is substituted instead of stop codon, so a full-length RF3 is produced.